Welcome everyone to our eSTAR webinar today. Just a few in background. So this is a Teams meeting. So we do want engagement after the presentation, but please mute yourself until after the presentation's over. We also ask that while the presentations start, not to come on camera. After the presentation for the Q&A, folks can feel free to raise hands or put things in the chat throughout the presentation. There are closed captioning available. There's a way to turn that in kind of from the different dots. If you have any technical issues, I'll be on my email so I can try and help. So please just send me a note. And then as we go, people can also give kind of thumbs up to questions we want to definitely answer. So this takes place within HESI, which is the Health and Environmental Sciences Institute eSTAR Committee. And that's our Emerging Systems for the Toxicology for the Assessment of Risk. And I won't read this line by line, but really our mission is to just develop and deliver innovative systems toxicology approaches for risk assessment. We have a portfolio of 6 very active projects that all focus on different omics technologies to really use it for decision making. So beyond just understanding mechanism, but really how can we use this to answer different scientific questions, mostly related to toxicology. With that, we have our webinar today. I'm really excited to welcome Dr. Jesse Salk. I've gotten to work with him the past 4 or so years. It's been really great getting to know him. Jesse Salk is a clinical oncologist, a molecular biologist, and the founder of Twin Strand Biosciences. Oh, but a little bit of feedback. If you aren't talking, please just go ahead and mute yourself. Okay, should be okay now. He co-developed duplex sequencing and error-corrected NGS technology being used in a variety of collaborative studies across HESI, including GTTC, CT-Trex, and eSTAR that I just mentioned. He works with a variety of companies on new genomic technologies and sees cancer patients part-time at Seattle's Veteran Hospital in Seattle as a clinical faculty member at the University of Washington and Fred Hutch Cancer Center. Today, he will be discussing a new spatial DNA sequencing technology and its potential applications for early carcinogenesis assessment via in situ detection of cancer driver genes and mutation-bearing clones. All right, thanks so much, Jesse. I'll stop sharing and the floor is all yours.
Hey, thank you. So we got a lot of people. So today, a few things. I have 2 slide decks I'm going to go through. I'm going to switch one in the middle because it's going to prevent some funny business that was going to happen if I merge them. I also know we got a lot of folks on here, and there are a few of you I owe responses to in different things. So I'm just going to give you a bulk apology. I'm going to try to catch up on that in the next couple of days. I'm going to spend the first part of the talk trying to give a little bit of a background in some of the context related to some of the projects that have been going on with eSTAR. I'll touch on a little bit that relates to some of the other groups within HESI. And in the second part of the talk, I'm going to really focus on this specific technology that is sort of representative of a new class of technologies that I think are going to be particularly relevant to eSTAR and across HESI. So to begin, and some of these slides will be familiar to some of you from different conferences and presentations before that we've done together. But as initial framing thing, I think is a little bit funny, but also kind of an interesting way to think about genetics. So I'm a molecular biologist focused on genetics and nucleic acid and how it affects human health. And if you think about before the human genome was sequenced, we had a variety of technologies for sort of low throughput looking at genetics, sequencing individual genes or looking at individual chromosomes. And then once we had next generation sequencing technology in sort of the early 2000s, the first after genome in the after genome epoch. The first sort of era was focused on the inherited genome. What makes one person different than the other? Why some of us have blue eyes, brown hair, risk for particular heart disease, et cetera. And we sequenced a lot of different individuals and looked at variation between them. Then the sort of next thing that was focused on really was the cancer genome. And this was spending a lot of time sequencing a bunch of individual cancers of different types and different individual patients with those different cancers, seeing what makes them different or similar and ways that we can kind of treat that. And although all these genetics and genomic technologies are used together, I think thematically, that's kind of a fair way of looking at it. And I'd say we're in the beginning of what I'm sort of foreseeing to be the third era in the after genome epoch, which is the somatic genome. And what I mean by the somatic genome is the genome that is not the one that was present in the single fertilized zygote when we were, conceived, but that develops in the body after later in life. And if you think about the human genome, we typically cite it as it's a 3 billion base pair genome. You have two copies of it, one from mom and one from dad. So each cell has 6 billion letters. But if you really think about what the somatic genome is, it's the multiplication product of the number of cells in the body times the size of the genome, because every cell is actually a little bit different. And the size of the human somatic genome is actually 420 sextillion base pairs if you multiply 6 billion times 40 million cells in the body. So that's a lot of, it's a big challenge, a big area of technical complexity. Things within what I call somatic genetics include things like embryonic mosaicism. There are mutations that accumulate early after fertilization that occur in some cells and not others and spread to a subset of cells. There are probably some unsolved neurosyndromic cases, patients that don't have anything obvious in whole genome sequencing, but they have mutations in maybe a subset of cells in their brain that cause a phenotype. That's an example of that. Adaptive immunity, your B and T cell receptors rearrange, and that is somatic genetics, and that is necessary for us to survive and evolve resistance to microbial organisms. Mutagenesis, things that we're exposed to during life that happen either naturally or through chemicals that modify our genome and mutate it, that's somatic genetics. Preneoplastic clonal evolution, a lot of what HESI's eSTAR is focused on is somatic genetics. Cell transplant and cell therapy is taking cells from another person and putting them in your own body, or modifying cells and putting them back is somatic genetics. Interventional genetics, so gene editing and things along those lines, where we're intentionally modifying cells therapeutically and sometimes with unintended collateral consequences, is somatic genetics. And one other category, I think is also arguably somatic genetics, is the microbial genetics in our body. We have more microbial cells in our body than human cells, and these are essential for a lot of aspects of our physiology. And so these are aspects that are not things that we inherited when we were conceived, but have accumulated or changed during our life. And some of these should be very familiar to many of you. Mutagenesis, preneoplastic clonal evolution, and aging, and cell and gene therapy are in fact the backbone of the GTTC, eSTAR, and CT-TRACS groups within HESI in terms of how these affect human health and safety. Again, many of you have seen this slide or derivative of the slide by me in the past, sort of talking about somatic genome safety. One of the major things that we think about with somatic genome safety is cancer. Things that mess with DNA can increase cancer risk. Things that create mutations or let cells that have mutations sort of grow in an abnormal way, and whether those mutagens are non-specific things in tobacco smoke or targeted agents like CRISPR, they're modifying our genome, and that has a risk of leading to cancer long term. But of course, there are other things that genetic modification can contribute to in terms of health consequences: reproductive, genetic diseases, autoimmunity, a variety of other things. But cancer is kind of the main one we mostly think about. Things that modify, things that are, can cause cancer, we call carcinogens. And there's a variety of types. There are some that directly mutate DNA, and that can make cells grow in an abnormal way. That can be a carcinogen. So mutagenic carcinogens are one type of carcinogens, but there are other types of carcinogens that don't directly mutate the DNA, and they just let cells that have pre-existing mutations that have accumulated through natural means preferentially grow out, and those we call non-genotoxic or non-mutagenic carcinogens. And then there's a special subset of mutagenic carcinogens, which are very targeted agents, things like CRISPR, that we intend to have modify one part of the genome, but can have off-target effects. And again, sort of eSTAR, so CT-TRACS, focusing on gene editing, safety, GTTC, looking at typically kind of mutagenic agents, largely focused on non-specific acting ones. And eSTAR has been mostly focused, at least in the DNA sequencing genomic technology side, on looking at non-genotoxic carcinogens, things that can facilitate cells growing when they shouldn't, but that are not necessarily directly mutagenic. So that's a little bit of the hierarchy. In all of these groups, I developed a technology called duplex sequencing, which is one of the error-corrected sequencing methods that are being used by these different projects in these three groups, and many of you are seen or involved with. The general concept is making multiple labeling molecules in a way that they can be uniquely distinguished from others, making multiple copies of those and sequencing them, and then knowing that even though there could be some errors in some of the copies, if you sort of create a consensus, you group them all together and you average out anything that's different from those single molecules. What started from a single molecule, you can remove errors, and duplex sequencing is one method. There's other methods that are being used, the PacBio platform and some others that are starting to be used across HESI, but that's a fundamental concept. And so, measuring somatic mutations or somatic genomics is tricky because it's only in a small subset of cells or molecules. It's not in every cell. And to normally be able to see something present at 1 in 1,000, if your error rate is 1 in 100, you can't see it. You have a signal to noise ratio problem. And these error corrected sequencing methods have allowed us to drop our error rates enormously and detect these very low frequency events, which is awesome. And it's made some real neat data across the 3 different HESI groups. And one example with duplex sequencing in the far left side here is showing, this is small region of the genome, 250 bases. Each tick along the axis is a nucleotide position. And in this case, the y-axis is the percentage of molecules or percentage of sequence reads being sequenced that have a variant in it, and it's zoomed into 1%. Standard sequencing shows apparent mutations at low percentages, sub 1% in essentially every site. That's all technical noise, that's background errors. Duplex sequencing, one of the error-corrected sequencing methods, same exact sample, gets rid of that background noise. You can see that one true mutation that's present. And that has been really enabling for detecting low-frequency mutations. But one of the things it doesn't do is let you know exactly where they're coming from. We take a piece of tissue, we grind it up in a bunch, all the molecules get mixed together, and although we can pull out that faint signal, we don't know exactly where it came from. And biology is rooted in structure and orientation of cells, these cells as these units. And it would be really nice if we were able to do this in a way that you could actually know what cell that molecule came from, what the pattern is, how cells with the same mutation relate to each other. So one of the challenges of using error-corrected sequencing in carcinogenesis assessment, which is what we've really been doing a lot with the eSTAR group. I'll show you a couple examples. One, this is a study that was published a number of years ago. Clint Valentine was the first author. We took some rodents that are these TGRAS-H2 animals. They have 4 extra copies of a human proto-oncogene of HRAS. And these animals are cancer predisposed, and the thought is sort of that part of the reason for this, they just have multiple copies of these extra RAS genes that have a 4 times more chance of getting randomly mutated and activating and kind of growing and letting tumors form. We used duplex sequencing to target the human HRAS proto-oncogene as well as some endogenous RAS genes in these mice, and treated them with urethane. In this case, urethane causes lung adenocarcinomas and splenic hemangiosarcomas in this model within a fairly short time, about 6 months, cancer accelerated model. And what we were able to see using duplex sequencing is that during this, within about a month after exposure, there's a few random mutations scattered across some of the genes, but you'll see these subclonal mutations, things not present in a single copy, but present in 1% or 2% or 0.5% of molecules in the target organ. So we could see preneoplastic clones in histologically normal lung and spleen. Those clones, and those are the 2 tissues that develop these tumors. And we didn't see it in other sort of non-target tissues. And this is pretty cool, saying we can see early emergence of preneoplastic clones, things that are destined to become cancer on their way, but in morphologically normal tissue because we're able to drop our error rates low enough to see these subclones. So super cool, very promising. Another study we did with someone at UCSF, Alan Balmain, very well-known cancer biologist, used a classic skin painting model where you can either treat rodents with nothing, with painting on a tumor initiator called DMBA, or initiator followed by a promoter, DMBA followed by TPA. And in that model system, if you do the initiator followed by the promoter, you get skin papillomas, 6-8 weeks after this treatment. And if we know that those papillomas are driven by specific mutations in RAS genes, and we can see if we use high accuracy duplex sequencing to look at the hotspots of these RAS genes, in the vehicle or initiator only, we don't see these little microscopic clones, but you can definitely start seeing them in histologically normal tissue. In histologically normal tissue after about 6 weeks. And the tumors that these animals get actually show up at 4 to 6 months. So we're able to see tumor precursors based on genetic lineage tracking of cancer driver mutations in histologically normal tissue. One case showing a mutagenic carcinogen, another case showing an initiator followed by a promoter, which is a promoter is a non-genotoxic carcinogen. And there's a few other studies that have been presented in HESI and some of the members, and there's an active error-corrected sequencing working group within eSTAR planning on how this could be broadened in using this technology, duplex sequencing, to pick out these clones. Really cool. But as many of you know who have been part of that working group, there's some challenges to doing this that come down to some practical things. And I've made a conceptual little diagram here that I think will be a little bit helpful. So let's just say I take a piece of tissue, I extract DNA, I do crazy high accuracy sequencing, and I'm able to pick out a 0.1% clone, means 1 in 1,000 molecules have a particular mutation. Standard sequencing wouldn't let me do that because the error rate would be too high. But what does that 0.1% clone mean? Well, it could be that there's one giant clone plopped right in the middle here. That's example A. It could be that there's 5, 6 independent, equally sized small clones scattered around, or it could be that there's some small clones, some big clones, and if you add up all the mutant molecules divided by the total number of molecules, you get the same frequency across all of these. Biologically, it's probably relevant to know which is which, and we can't do that right now without that spatial information. Is it the questions, is that 0.1% mutation for one clone or many? Are the clones uniform or heterogeneous in size? Where are the mutations? Are they in epithelial cells, or are they in lymphocytes that happen to be floating through? Are they in terminally differentiated cells that can't turn into cancer because they're just going to slough off and die? Or are they in stem cells? Are they in a normal stem cell structure, like a crypt in the colon, moving from the bottom to the top? Or are they starting moving and migrating? Maybe morphologically they don't look abnormal, but based on the lineage tracing of these mutations, are they kind of displaying a weird behavior? Are the cancer driver mutations, that if we look at a few different genes, are they phased together in the same cell, or are they in different cells? Are there other accompanying molecular changes, like changes to transcriptional profiles or protein expression or whatnot, that are different and accompany these mutations? And what is the natural history of all of this stuff? The reality is we know that these type of clones occur with normal aging, and we know they seem to occur at accelerated rate, these clonal outgrowths, with carcinogen exposed animals and people. What is this sort of overall natural history of all these features? How is that different between sort of normal aging and carcinogenesis? And just getting that number 0.1%, it's great. We haven't been able to do anything like that before, but it's really incomplete if you think about how you answer these kind of questions. And so that's one issue. From a practical matter, again, those who've been in the working group within eSTAR, we've been talking about taking some animals, treating them with non-genotoxic carcinogens, and picking a panel of common cancer driver genes that we think are potentially mutated in the tumors that form from those animals, and looking at early time points. And we talked about how many tissues do you sample, which tissues, what is the size of the piece of tissue. And it actually matters, and this is sort of tricky. If I take an entire chunk of tissue and I grind the whole thing up, I've got 100 million cells in there. If I have a clone that has, let's say I have a piece of tissue is 1 million cells, and let's say I have a clone of 10 cells. If I ground the entire thing up, that would be a 1 in 100,000 variant allele frequency. And although error-corrected sequencing can resolve things at that level, it becomes very complicated and expensive because you have to sequence extremely deeply to have a high probability of detecting 1 in 100,000. You need to sequence millions of molecules. If you sample smaller pieces of tissue and you say you punch out 10,000 cells and your clone is 10 cells, you're going to see a 1 in 1,000 clone. You don't have to sequence as deeply, but how do you know you got lucky and you happen to sample right around that clone? If you take a bunch of small pieces, some of them might miss that clone, some might catch half the clone. And the more of these samples you take and the more the clones you are, the more probability you have, but you have site to site variation. So it's a great technology. It's shown a lot of promise and something enabled us to do things that have never been possible before. But in theory, wouldn't it be nice if we could just look at this picture and say, where are those cells? Like not in a test tube, but in the actual tissue. So that's the technology and concept I want to talk about today. I am going to switch over to my other deck. Okay, and I will apologize that some of the, I've tried to clean this up, but some of these slides may have a slight commercial bend to them, so just ignore that. Okay, you guys can see this hopefully.
Yes, looks good.
So there's a company I've worked with for quite some time called Singular Genomics, and I'll tell you a bit about them and this sort of new platform. But what I want to kind of preface this by saying is just like I said many times to others, in talks, that science requires all sorts of different tools. No single tool is ever going to answer all the questions. And we're only as good as our tools, but we also can't just be wedded to any single tool as the end all and be all. And so this is a platform that is what I expect will be the first of a variety that will come out over time. Just like duplex sequencing is a very good version of error-corrected sequencing, but there are others. So think about what I'm going to talk about with this platform in the context of what's available now, but also kind of a general phenomena that I expect will be kind of an era of science we're moving into. So Singular Genomics is a company that started focused on making a sequencer box to kind of compete with Illumina, to make a sequencer that does similar things. You extract DNA, you process, you make libraries, you put it on the machine, and the machine's a nice size, it's flexible, it runs a little bit faster. But the basic sequencing concept was fairly similar to other platforms. What they've spent time doing over the last couple of years, which was always sort of part of their plan, but only kind of was announced recently, was to take this sequencing platform and actually turn it into a spatial genomics platform, which can essentially do the thing I just described, actually resolve in single cells in the context of a piece of tissue exactly where these molecular changes are occurring. If you think of pathology historically as, you can be board certified in human medicine in what's called anatomic pathology or clinical pathology. And many people, are board certified in both. Anatomic pathology means like autopsies and looking at stuff through a microscope. And clinical pathology or molecular pathology is these sort of fancy molecular tests. They're kind of different specialties. The idea is you can't see certain molecular things in a normal tissue section. With molecular methods, you're extracting molecules and doing fancy things, but you lose that morphology. Wouldn't it be amazing if you could just bring those things back together and not have to have a trade-off? And that's kind of what the idea here is. So the way this apparatus works is it allows one to look at small sections, thin sections of tissue, formalin fixed paraffin embedded tissue, routine clinical type of samples, and get 4 different types of readouts. Sequencers are basically just a really high-end microscope that scans across a slide and it has the ability to read out fluorescent signal in a fast way, and microfluidics that allow different chemistry to be flowed across. So a sequencer is actually not that different than a microscope. Much of what's in a sequencer is a microscope. So what this platform allows one to do is four things. It allows one to look at morphology and look at effectively a standard H&E image. It allows one to resolve multi-analyte proteomics, so looking multiple types of proteins expression at the same time. It allows one to look at multiple transcripts at the same time. And the really novel thing that's going to be particularly relevant to this group is the ability to do direct sequencing. There are a variety of companies out there that are spatial technology companies or spatial sequencing, NanoString, 10x, Akoya, a variety of others. And most of those are really focused around transcriptomics or proteomics. And the thing that's quite novel about this apparatus is the ability to do nucleotide level sequencing in situ. And I'll show you why that's relevant to you guys in a minute. And it's not that these are done in serial sections, you know, of you cut this, you do that, you cut this, you do that. They all are done in the same exact tissue section, which is quite important because it allows one to look at literally the same cell and say, what is the sequence of a given gene in that cell? What is the level of transcription of a bunch of transcripts, proteins, and where does that morphologically sit relative to other cells? And look at the exact same cell for all those three things. So, four effectively different readouts: RNA, protein, fluorescent, H&E, morphology, and sequencing. And how does this work? Well, all of these things are, well, not the morphology, that's just effectively H&E, but the three other things, transcriptomics, proteomics, and the nucleotide level sequencing are actually all sequencing based. The way this works is you take your 5-micron section, you put it on a slide, and you build the flow cell around it, basically. In the context of making this, the cells are fixed, but they're kind of partly permeabilized. So there's lots of little holes in them that actually reagents can get into. The RNAs and proteins and DNA is stuck inside the cell. It's kind of locked in there and it can't float away, but little bits of sequencing reagents can actually get in. So each cell is like a little bag. And with a typical sequencing library, you have molecules that sort of stick on a surface and get clonally amplified, something called cluster amplification. And this is happening the same thing here, but it's inside the cell. So your sequencing clusters are actually inside the cells. And so for transcript counting, the way this technology works, there's special targeted probes called molecular inversion probes or padlock probes that have a couple of feet that bind to a target sequence. In this case, in the upper left, I'm showing you pink and blue. Those are targeting arms are about 15 base pairs. They specifically bind to a sequence, and they have a linker, a black region between them. Those two ends of the pink and blue are in close proximity. And after these probes are annealed, a ligase is put in there, DNA ligase, that if those two ends are close together, which they only should be if they're bound, they will ligate and create a little circle. And then one can use what's called rolling circle amplification to create these concatenarized molecules that kind of collapse down on themselves and as little dots, which they call, in this case, what do they call it? RCPs, rolling circle products, but they're little clonal clusters from a concatemer. And then sequencing is done on that. The direct sequencing is the same exact process, but there's a gap between the 5' and the 3' arms. And so before ligating, you do an extension. And this is actually a DNA-based probe going to an RNA-based target. So this is looking at transcripts. One uses an engineered type of reverse transcriptase to extend across, bump into the other side, fall off, ligase circularizes it, and you do this rolling circle amplification. But because you filled in across this variable region, the region you're looking to sequence, for example, an exon of p53. You want to know the sequence of a cancer driver gene, or sequencing across a T cell receptor, or sequencing across a SNP site that's different in a donor cell from a recipient cell. You can get the genotype information incorporated into that concatenarized cluster. And then for protein detection, it's the same concept as the sort of transcript counting, but you have antibodies, typical immunohistochemical antibodies, but instead of having a fluorescent dye on them, they have an oligo with a unique barcode, and one is just directly sequencing the barcode. So sequencing is the ultimate basis for reading out all three of these things, but you're measuring three independent things. And able to, again, they're all on the same section. They're happening in the same cells. One does one, washes out the reagents, does the other, washes out the reagents, does a third. And the sequencing process is exactly the same as this normal sequencing process. Instead of clusters on a glass surface from an extracted DNA library, you have clusters that are stuck inside cells that the clusters can't get out of, but the sequencing reagents can get in. So you use a primer that is sequencing these products, and this little spots inside the cell, these clusters, if they're, if there's an A incorporated, the spot turns red. If there's a C incorporated, the spot turns green. If there's a G incorporated, the spot turns yellow. And if there's a T incorporated, the spot turns blue. And so essentially, you're getting clusters inside cells that look something like this. This is zoomed pretty far in. This is showing one cycle of, this is zoomed way, way, way in a piece of tissue. The red outlines are a computer looking at the image, because it's done fluorescent imaging of H&E of these cells to begin with. So it knows, it can sort of say this is the outline of a cell based on either staining the nucleus or using a protein stain in the cell, or just extrapolating that from H&E. And it says, okay, I as a computer think everything inside this this red bounds is a cell, and every spot, every cluster inside that cell came from a single molecule that is stuck in there, basically. And so I'll show you in a minute what it looks like with the sequencing, but each cycle, each spot turns a different color. And so the ultimate readout you're getting normal sequencing readout. You're getting FASTQ. Each one of these clusters is AGT, CCGT, whatever. And each of them has a quality score, each letter, for those of you who are familiar with a FASTQ file. But each one of these spots also has an XYZ coordinate. And yes, there actually is a Z coordinate. They image that 5 micron section, a few different layers. And because you've outlined the cell, you say, okay, this exact spot has this exact sequence, and it has this XYZ coordinate, and that XYZ coordinate falls within this boundary of a cell, and so it's in this particular cell. And so cell 1 happens to be next to cell 2 or cell 3. And so it's really just sequencing, but in this architecture. So this is zoomed out a bit, and this is showing you an example of looking at a handful of transcripts in a tissue. You're seeing this video of these spots, which I know are really small because, again, this is zoomed out a little bit. You can't see each one very clearly, but you see how the colors are changing. You can also see other sort of patterns of like these little loops and zigzags and whatnot. You can see there's some structure in here, and that's because this is from a human kidney. And the reason you can sort of see those structures, and in some cycles you see a green outline around one thing, that's because all those clusters are turning green in that cycle, because those are all a particular structure in the kidney, the glomerulus, the collecting duct, or whatever it is, that happen to have that particular gene being transcribed in those and not others. So there is far more information that comes out any given cycle of this than you can really understand or interpret, visually all at once in your head. But this is kind of what's happening at a deeper level. If you look at a handful of transcripts, in this case about 100, by eye you can't just sort of deconvolve all the A's and G's and G's and C's yourself. But what you can do is you can say, well, of these 100 transcripts one was looking at, these ones showed up in this cell or that cell or this other one. And you can say, well, we happen to know for historical reasons that this pattern of transcripts corresponds to proximal tubule cells or renal corpuscle or glomerulus or whatever. And you can then create a pseudo-colored image that you just said, I'm defining each one of these cells, I'm going to put a pink spot on it if it's a proximal tubule cell, which I've defined based on having certain, transcripts present, for example. So you can, pseudo color, you can put things in an image that, a human can understand by just sort of adjusting your, criteria for how you define those colors later. Same thing can be done with proteins. This is looking at, you remember antibodies with oligos that are then sequenced. This is doing a few different rounds of sequencing, 4 different antibodies each, CD8, CD4, CD3, et cetera. This is from tonsils. People often use tonsils in these examples because they just create these pretty pictures. But you can see, showing 4 different proteins with four different colors at a time, but this is the same exact tissue section. They're being shown separately to make it easier to visualize, but it's a 8-dimensional color scheme here. And so again, to put this in the context, this is a 10 by 10 millimeter section, 5 microns thick. So 10 by 10 millimeters is 1 square centimeter. So I'm trying to zoom in and out so you kind of get a sense of scale. So imagine, 1 square centimeter, your thumbnail size. If you zoom in a little bit, I've zoomed in, about 100 times here, you can get a sense of, these different structures. This is based on pseudo coloring based on the transcriptional profile. Tubules, glomerulus, collecting duct, et cetera. This is still zoomed out about 100 times further than the sort of single cell level with all the little spots in the transcript. I'm just trying to give you a more visceral sense of this sort of dynamic range, zoomed in, zoomed out. Okay, so now I'm zooming back out, to this piece. Now imagine that you take 10 serial sections. You cut 10 different sections that are immediately adjacent to each other. The way this machine works is it has these flow cells in it. They can handle 10 square centimeters each. You can actually take 10 of these sections. They could be from the same serial sections and Z stacks. They could be from totally separate samples. And you can fit 10 square centimeters on here at a time. You could use a larger, you could do 10 1 centimeter by 1 centimeter, or you could do, 40, 50 smaller pieces. And so what you're effectively getting is, a surface area, and you have, that much to work with per flow cell. And you go through these different cycling parameters. You can look at the morphology first, the protein second, the transcripts third, and the immunohistochemistry and the sequencing fourth, but they're all essentially the same thing. And the machine actually can handle 4 of these flow cells at a time. So you can actually get basically 40 square centimeters of tissue you're working with. And if you think about, how many cells that is, that's an extraordinarily large number. This is just showing some more human tissue. This is what the H&E looks like, fluorescent H&E, standard, stains for slides. This is looking at 10 proteins at the same time from that same tissue, and this is looking at 150 transcripts. And again, this is zoomed way out. You can zoom in 100 to 1,000 times more to get down to that subcellular resolution, but there's a lot of information in there. And again, you can do this thing called UMAP scores, where you kind of group cell types by different patterns of proteins or transcripts, for those of you who've done spatial genomics, and kind of put that together and color it however you want to look at it. So that stuff is all well and cool, but that stuff is similar to what some other spatial platforms can do. The thing that's super cool and relevant here that I want to really focus on is this direct sequencing. So this is examples. This is not from tissue. This is from cells sort of spread out on the surface and fixed down in culture, but it's the same thing of looking at a CRISPR edited population. Showing you the same cells staying with fluorescent, with H&E or a couple of proteins, and then actually going through the cycling of sequencing that actual CRISPR product. These were edited cells, and you can see those white outlines define the cell boundary, and you can see there's multiple transcripts, multiple spots per cell, because one integrated gene can make multiple transcripts. And you can see that some of the cells, during some cycles, they all are the same color, but that one in the upper right looks different during some of the cycles because the integrated gene is different. And so this is interesting, kind of to think about consensus sequencing and error-corrected sequencing we talked about in the past. The way, remember, error-corrected sequencing, duplex sequencing, the way it improves accuracy is it looks at multiple copies that you biochemically made of the same starting molecule. The underlying sequencing is no more accurate, but you're creating this labeling system to create a consensus. And what this is doing, although the sequencing is no more accurate, what you have done is you've taken one molecule, you have concatenarized it to make a colony, and you also have multiple copies of different transcripts per cell. And so even though any given sequence read could have an error in it, you have multiple copies at multiple different levels. And so whereas like duplex sequencing is using molecular tagging to create consensuses when you have distributed reads, what you're able to do is create a consensus based on the spatial coherence in this case. And so although the accuracy on, again, per sort of extension reaction is not 1 in a million, the fact that you can look at multiple transcripts per cell, which are biochemical copies, and you can look and say, oh, this cell is next to this cell, is next to this cell, maybe they're clonally related, that actually gives you the opportunity to sort of do some of that same kind of concept of error correction. So that's one example. This is another example. This is looking at, it happens in B cell receptors. This is a mixture of different, standard B cell lines, lymphoblastoid cell lines, and you can see sort of cycling through the colors of these spots are a bit different between cells, and that's because you're directly reading out the B cell receptor in this heterogeneous mixture. So pretty cool. You could apply this to any one of those things I talked about for, sort of spatial genomic or somatic genetics. But, what about like the eSTAR specific stuff? Remember, we talked about how do we go about looking for clones in a way that, we have to be sort of careful. How are we sampling? How are we sequencing? Well, this is just an early example. This is from some animals, some mice that were treated with that same skin painting model we talked about before, the initiator followed by a promoter. And this is looking in some of the actual papillomas that form, a number of months later. And this is just looking in a couple of the RAS genes. This is looking in one RAS gene. And let me walk you through how to sort of interpret this, and this is not the end all and be all, but it gives an example of what the concept is. So mutations in RAS drive most of these papillomas. So I'm showing 4 different spots representing 4 different colors, 4 different colors representing 4 different genotypes. The blue and the red spots are looking at the codon 12, and the green and the yellow are looking at codon 61. Blue is wild type, red is mutant for codon 12, and green is wild type, yellow is mutant for codon 61. And then the far left, I'm showing you just one example of just a piece of normal skin, and all you see are blue and green spots. So wild type for both of those 2 things, both those 2 loci. In the middle panel, which is the benign papilloma, you are seeing blue spots and some green spots. Those are wild type for those 2 codons, but you're also seeing yellow spots. That is a mutation in codon 61. And remember, since this is an oncogenic mutation, it will typically appear in one allele and not the other. So at codon 12, you have wild type because that's not involved in this process. At codon 61, you have one allele that's mutant and one allele that's wild type. And this pattern makes a lot of sense. You don't really see any codon 12 mutations, no red spots. In a totally different papilloma, what you're seeing is different. This other papilloma is driven by a codon 61 mutation. And so a codon 12 mutation. And I know that red coloring here is really hard to see, but the point is there's not yellow spots, there are red spots here. And so as you would expect, one papilloma that's driven by a particular mutation will have one, but not the other. And this other papilloma will happen to have, these are two different ways of activating this gene. But the point is you're seeing clonality in a situation you exactly expect it. So the next step for this is to say, well, what if we look in these, histologically normal tissues? Can we pick out small numbers of clusters of cells that have this mutation or that mutation, or multiple clusters of mutations? Do we see some clones with codon 61 and some with codon 12? If we look in some of the model systems that the project that has been done with Merck and Taconic and Charles River and TwinStrand and Pfizer has presented to this group, looking in benzodiazepine treated animals or PCB 126 treated animals. Can we find some of the mutations that you would expect in those in small subsets? And so these are just very small piece of tissue, early example data, but showing proof of concept. But one can, a given sequencer run, look at hundreds of times more cells than this. These are just tiny little spots. A couple last things about this, platform. The things that are truly different, really differentiated about this apparatus, which is sort of announced but not fully commercially released yet, are one, it can do the direct sequencing, which is very, very novel. Second is its throughput is much higher than other tools out there. It can do 40 square centimeters at a time as opposed to a few square centimeters, and it takes about 24 hours to run as opposed to in some cases several days. So the throughput is much higher, so the costs are lower. And so, one can, like I said, you could take some modest number of large pieces or a large number of small pieces, and you can run up to, I don't know, something like 100 or 150 samples on a given run if one's looking at small pieces, which really, has a lot of potential if one is thinking about looking at the number of tissues and sizes and that we've sort of talked about as being sort of ideal. So that's kind of the gist of it. It takes, runs in the sequencer run itself is about a day, but it takes a couple of days to sort of prepare the sections. The neat thing about the way you load these slides is you actually can take little punches out of, you cut a section on 5-micron section from a block. It floats in water like you normally do, but then you pick it up on a slide that has some hydrogel on it. Think of it like acrylamide or agar, something a little bit squishy, and then you use a fancy cookie cutter to punch out the areas you want, and then you flip those over on the flow cell where you want to put it, and you peel off the hydrogel, so your really thin, delicate little layer isn't disrupted. It's protected by that hydrogel. And so again, it's easy to kind of mix and match and kind of prepare one sample at a time and kind of load them on this way, which is pretty cool. So kind of last slide in summary, this apparatus takes standard 5-micron sections, gets morphology, transcripts, not the entire transcriptome, but a relative, modest number of defined transcripts, 200, 300, 400, a few dozen proteins at a time, and can probably do 10, 20, maybe 50 kb of direct sequencing of looking at variable regions. Can look at about 40 square centimeters per run and do about 24 hours at a time. And I think the neat opportunity with an eSTAR, lots of opportunities in other working groups too, but specifically for some of the projects that are ongoing related to cancer driver mutations and some of the duplex sequencing panels we built, to take some of what we learned from those studies, use the contents of those panels to build an assay on this apparatus, and then use the corresponding FFPE blocks from these tissues to be able to see if we can, identify the in situ distribution of these clones, and then, potentially look at other molecular phenotypes too. What types of cells are they in? Are they transcriptionally, looking abnormal? What is the protein expression, et cetera. So I wanted to let people know about this and sort of bring it up as an emerging technology to be aware of and discuss. So that's it. Questions?
Great. Thanks, Jesse, so much. So folks can either do a raise hand if they want to ask their question and unmute, or they could put things in the chat and I can moderate. Jesse, maybe while we're waiting, I know you had talked about maybe applications for things like CT tracks. Do you want to talk briefly of what that might be?
Yeah. So, I mean, imagine what you could do with in terms of direct sequencing. So let's just say I had a patient who had leukemia and I treated them with a bone marrow transplant. And then I followed that by treating them with a CAR T cell. You could identify which cells are the cancer cells based on the specific mutations that are in them. You could identify the donor cells from the recipient cells based on looking at different SNPs, and you could identify the engineered CAR T cells looking at the integrated cassette. And you could say what's doing what. Is this causing graft versus host disease? Is this causing graft versus leukemia? Is this CAR T cell working? Is this not? So within CT tracks, both the kind of biodistribution, seeing where these cells, engineered cells go, whether they're autologous or allogeneic, and then also thinking about in situ gene editing. You deliver a nanoparticle carrying a CRISPR system or an AAV or something. You could see what cells are edited. You could look at on-target sites. You could look at suspected off-target sites. With the CT tracks group, some of the early work we did, we saw that there's different cell types in culture, at least. The off-target pattern actually seems to depend a little bit on the cell type. And when editing in vivo, I mean, there's a lot of cell types in there. So yeah, that's that one.
Great. Yeah, cool to see the different applications. Bevan asked a few questions. The first is, how many different transcripts can you analyze in one single run?
Right now, I think they're sort of saying somewhere in the order of about 200. That's not, an absolute limit. The things that sort of, because you're not labeling a bunch of individual probes and having to deal with color space, you're getting sequence reads. You don't have a kind of a informatic limit. Your two things you're limited by are really, one, how many of these little balls that you can fit per cell is your steric crowding, and that's actually not limiting. It's what's the resolution for how many balls can you kind of image at the same time before they overlap. But what you can do, you make all of these little clonal clusters, and you can put in primers that just sequence half of them at a time, and you strip off that primer and do the other ones. You can kind of space things out. If there's some transcript levels that are really, really high and others are really low, there's a little bit of complexity to that, but it's probably on the order of a few hundred now and probably a few thousand long term.
Great. And she also asked what types of tissues or organs have been validated so far.
Yeah, I think it depends on exactly what you mean by validated, but bone marrow, kidney, tonsil, colon, in human, mouse, skin. We've got a few others. I've got a bunch of other sort of pilots ongoing with some different academic groups, but it's pretty universal. Question about, is this applied to archival FFPE blocks? The answer is yes, and that's really interesting and exciting about this. So I didn't show you this data, but one of the early data sets was from bone marrow from 12-year-old bone marrow core biopsies. So they're old, but the thing about bone marrow in humans, you don't deal with this in animals so much. In humans, you need to decalcify these so you don't dull your blade because of the little spicules of bone, and that tends to be very damaging using formic acid typically. This actually worked quite well in formic acid decalcified bone marrow that was 12 years old. So this opens up possibility of historical samples. It may be a little bit harder for the direct sequencing because you have to fill in a gap rather than just ligate it, but it seems to work pretty well. Question, Jake. Okay, let's see. Somatic variant may be present only one or two chromosomes, and there may be some ploidy abnormalities. How do you interpret that when you have aneuploidies? Great question. This is coming off of, so a couple of things. So this is coming off of transcripts, not directly off of DNA, and there's a couple reasons for that. One is with DNA, you only have 2 copies per cell, and if you this is not a perfect conversion. So if one of them doesn't successfully convert, you sort of miss it. Something that's heterozygous would appear homozygous or hemizygous. And with the transcripts, you have multiple copies each. But you got to remember that any given transcript, there can be imprinting. You can have maternal allele transcribed, and the other one less so. In cancer cells, there can be aneuploidy. You can have multiple of copies of one or the other. You can have copy number, neutral LOH, or gene conversion. I think it's going to be a matter of kind of playing around and testing. I think in some cases it may be possible to phase the variants together on the same read with a SNP that's nearby to kind of distinguish some of that, but that will be a tricky thing for sure. For chemical induced mutations, we don't know the target region. It was a good idea. So yeah, and so I'd say for this technology, this would not be a good technology for general mutagenesis, because this is that will be randomly distributed in molecules. I mean, it's actually that's not completely crazy. I mean, it maybe could be, but I would really one could theoretically try to do this and use like a kind of like a human mutagenesis panel like we've done a lot in GTTC, and look for cells that show a mutation. And it would ideally, you would be looking for a mutation in multiple transcripts in a single cell. So you're kind of overcoming that background error rate by looking for that. My gut feeling is that's a bit far out. I think the place that this is going to be most useful is looking at cancer driver genes, where you're going to see clusters of cells, multiple clonally related cells sharing the same mutation in close spatial proximity with multiple transcripts per cell mutated. In the case of oncogenes, it's going to be half of them. It'll often heterozygous. Okay, so what is the lower limit of detection? Percentage clusters say noise, not noise. Great question. Do not know. I think it's going to depend on how many transcripts you get per cell. It's going to be the types of variants you're looking at. It will be tricky in that some transcripts are just inherently higher than others per cell. Another limitation of using transcripts is you can't look at non-transcribed regions. And that's another sort of limit why this wouldn't be great, sort of mutagenesis type stuff. But for, things where you have a focal region you're looking at and you're looking for, clonality, I think it's, really interesting, and we will have to see and do some really specific validation.
Sorry, Jake, did you want to respond to that?
Oh, sorry. I actually had a different question. I don't want to hog everything. But just if you could quickly say, like, what's kind of the practical process of going from you get your the sequencer does the sequencing via imaging, you end up with FASTQs, you go do variant calling, and then you come back and you say, I want to see which cells have a specific mutation. What's that kind of back and forth? How does that kind of back and forth process work?
Yeah, I mean, under the hood, each cluster is like a FASTQ. So it's got a sequence and it's got quality scores, and then it's got an XYZ coordinate. So the way that this would basically be done is you would say, if you defined a cell area, at any pixels within this area is belonging within a given cell, you just look for a given sequence read, a FASTQ mapping to a thing. In that cell, you could say, what are all the transcripts or sequence reads per cell, or you could sort the FASTQ by anything, any read that has a particular sequence. You go back and you see what's in the cell, and you can flag only cells that show this read with a intracellular VAF beyond a certain point, a certain number of counts or frequency within the cell, and I mean, you can imagine doing this in Excel. Obviously, you wouldn't do it in Excel, but it's really as simple as that. It's a FASTQ with spatial coordinates, and then you can annotate the spatial coordinates as either intersecting or not intersecting with the defines of a given boundary of a cell.
We're at noon. I know there's a few more questions. Jesse, maybe in terms of follow-up, is it are you the best contact? Do you have a contact you could share with me and I can send in kind of the follow-up from the webinar if folks are interested?
Yeah, it's probably me for now, and I would hand off more. I mean, this sort of kind of application stuff. So this company is, it's early access to doing stuff in-house only, and the thing that they're going to formally release as a product is going to only start with the protein and transcriptomics and morphology, not the direct sequencing. So this is pretty bleeding edge early access stuff, and I'm kind of helping them a bit with that. So if people are interested in pilots or talking about applications, yeah, probably me for now, then I'll pass it off.
Great. Well, thank you so much, Jesse. I'll send you the rest of the questions. Don't mean to skip them, but just want to be respectful of folks' times. So thanks so much for the presentation, and we'll follow up by email with everyone. Thank you all. Have a great rest of your day.
Thank you.
Thanks.